I'm gonna be on a conference call for a while.
So when are you eating, or did you eat?
I ate a little bit, but then more later.
Hi, Detlef. How are you?
Hi, Katerina. Good.
Yeah, thank you for opening up the room.
Of course.
Could you do me a favor and make me also a moderator? So you click on my picture, and then there are a few options on the bottom, and one
Yeah.
of it is so I
Yeah.
can take care of. Cool. Thank
Yeah.
you.
Got it.
How's your day going? How's everything in Germany, right?
Good. Well,
Good.
you know, the state of the world is not the greatest, but my own day has been pretty good.
Yeah, that's true. Yeah, my parents live in Germany, so yeah. Hi, Cecile. How are you?
Yeah. Hi.
Hello.
Thank you. Hi, Katerina. Hi, Dr. Weigel.
Detlef. Just what it
Dit,
says there, Cecile. I hope it shows my first name. So
yeah,
Yes.
Detlef. Yeah.
yeah.
Yeah. Welcome to Science Society.
Hi.
Yeah, thank you so much for making it. And yeah, it's a great pleasure to have you. I hope you'll enjoy it. We'll wait a few minutes so people have time to arrive, and then I'll introduce you to the audience, and then the stage is yours. We're talking about your great research.
Cool. Hi, Nick.
Hey, how are you? Thank you for joining Clubhouse.
And
And
there's Gray.
thank you.
Hey, Gray.
Everybody, I think I'm unmuted now. Can you guys hear me?
You're a bit quiet, Gray.
Oh, okay, let's see.
Yeah, no, that's better.
Okay, all right, cool. How's everybody?
Good.
Good. How are you?
Good.
And nice to meet you. And
Nice to meet you too.
through voice, but still.
Yeah,
Thank you for making it.
of course, thanks for the invitation. This is my first time using Clubhouse or participating in Clubhouse, so excited to finally break the ice.
Yeah, that's an honor. Thank you for being for for coming here the first time. I appreciate it.
Yeah, Gray actually has come up with quite a few really good ways to explain the work we did. I really like the metaphors that Gray has come up with. They are I use them a lot, Gray.
Yeah, I like yours too. I think there's lots of there's lots of nice metaphors to try to explain it, or at least it's helped me think about it too and understand kind of to conceptualize it. Yeah.
Hi, LT.
Hello, hello. You have the little party hat. I have a balloon. Hello, everyone.
Oh yeah, so how do I do that? How do I get a party hat?
You
So
don't.
oh, okay,
It's a
I
sign to
can.
you. It's a
Okay.
sign.
Yeah, it's when you're new to Clubhouse, so people know you're new, so they are like nicer to you, and
Mhm.
don't assume you know all the jargon, the Clubhouse jargon that actually exists. So yeah,
Nice.
that's what.
It's okay. I've been here for a year. I still don't know a lot of things. It's okay with or
Mhm.
without it.
So maybe can this what's the Clubhouse? What's the like 1 minute explanation of what Clubhouse is all about? Like what what's it all about basically?
So it's a audio social media. So it's a different way of interacting. So it's not text; it's audio. I think that's the biggest difference.
That's cool.
And yeah, so it gets more. I think it sticks more the conversation since it's audio and
Yeah.
kind of people generate friendships and things like that more easily since it's more personal, since it's audio, I think.
And it's not. I guess like the thing about text, like Twitter or something, it's like very. You only get like a few number of letters, so it's hard to like have like explain something complex, like mutation bias.
Yeah, it's very interactive and the retort is immediate. So yeah, you will
Hmm.
experience some interaction with the audience. It's like just a phone call to a huge number of audience. And when there is a lot of people in the audience. One of my friends told me during the early days of Clubhouse, it's just like experiencing 1,000 hugs. Yeah,
Hmm.
yeah. So,
Cool.
but for a niche community, it encourages discussion and also learning. Even from very technical or hard science topics for enthusiasts. So we really appreciate this app, and I hope you will get to enjoy it.
Hmm.
When you come back again, to be a guest.
Yeah,
Cool.
that sounds great.
Yeah, so I can start introducing you, and then yeah, I'm sorry for the background noise. Like the offices are so full everywhere because everyone is going back to work. So and I'm sitting outside, so it's in the city. It's
Okay.
loud. I'm sorry,
Nice.
No
but
problem.
yeah, okay. So yeah, again, welcome to the Science Society, everyone. So today we have our guest speakers, um, Detlef Weigel and Gray Monroe. And yeah, it's a great pleasure to have you. And just to give you some background information about our guest speakers, Detlef Weigel, he was an undergrad in biology and chemistry at the University of Bielefeld and Köln, and he graduated in biology. He worked back then with Drosophila neurogenesis, and then he moved to the Max Planck Institute for developmental biology in Tübingen, where he did his PhD, and then he began to work with plants during his postdoc research at Caltech with Elliot Meyerowitz, and yeah, that's where he started to work with the plant model Arabidopsis thaliana, and later on he was associate professor at the Salk Institute, and yeah, and now to give you some background information about Gray Monroe, he he did his bachelor in science in biology with summa cum laude at the Appalachian State University. He his work was about evolution of centromeres, and he did his PhD in ecology at Colorado State University. And and then he did a postdoc with Detlef Weigel. That's how they know each other and did this work together. And now he's assistant professor of climate adaptation and plant genomics at the University of California, Davis. So yeah, thank you so much. And both got a lot of awards, numerous awards. So yeah, it's a great pleasure to have you, and the stage is yours. Thank you.
Thanks for having us.
Thank you, Katerina. It's really fun to be here. Great. You want to start by going a little bit through the history, how you got into this, maybe, the work that you did as a doctoral researcher already at Colorado State, where you got interested in mutations that take out entire genes and that surprisingly can have very prominent roles in adaptation to the environment.
Yeah, sure. Yeah, so I guess I don't know. Yeah, I'm not sure if you guys have read the papers or stuff, but I can kind of give you a background on how this came about. So while I was a grad student I became, well, I guess we'll start with this. Mutations are changes in DNA, and they are the starting point of all genetic variation. So whether good or bad, all genetic variation all starts with a mutation. And in grad school, I got interested in mutations that actually break genes. So mutations that cause genes to stop working. And normally we assume that those are going to be harmful, but in some cases, it turns out, we were studying plants, there's many cases where breaking a gene can actually be beneficial. So this was kind of surprising and interesting. Um, and I've been looking at different show this pattern. And then this is kind of related to what started when I was a postdoc, with Detlef. Still interested in kind of mutations and genetic variation. And the project that unfolded was kind of an accidental discovery, basically. Never really intended to be studying how mutation rate differs between different regions of the genome. But
And Gray,
on,
maybe
yeah,
just to quickly
yeah,
interject there,
yeah,
I always say, if we had started with the hypothesis that essential genes mutate less than non-essential genes, I think everybody would have thought we are crazy, and I
yeah.
think we ourselves would have thought this is really a lunatic idea.
Yeah, exactly. The only way we found ourselves thinking that it was true is just because we accidentally found it, basically. Um, because it is surprising, because we've all been kind of growing up on the phrase that mutations are random, and they're not there's no preference for good mutations to happen, or preference against harmful mutations to happen. So, for example, genes that are the most important, we wouldn't expect them to mutate less by any kind of way. But it turns out, as with most things in biology, it's often more complex than we originally thought. And what we found is that the genome is regulating itself in a much more interesting and elegant way through molecular processes that's allowing for these cool mutation biases to exist. Um, so yeah, I guess that's kind of the connection where this started, because there was a graduate student in Detlef's lab, Tanvi Srikant, who's the second author on this paper that we recently published, and she was showing some cool data about how the genome is structured in a molecular way, and it kind of led to a little light bulb that went off that said, "Hey, I wonder if that has any effect on mutation rate." And after a little afternoon of doing some data analysis of some existing mutation data, we found this pattern that was really compelling and really strong. Of course, we were skeptical of it, and so for the next 3 years was essentially seeing if that was true, if this mutation rate bias was really what was going on, or whether it was some kind of artifact. And so for 3 years, we kind of dissected it and added lots of data and lots of people. This paper that we published has authors from, I think, 9 institutes in 4 countries, and so it's been turned into this big project, because we really wanted to make sure that we were, on the right track.
And Gray, I think what's important to mention also, that it was not exactly trivial to show this, because one cannot easily show this directly, because genes that are important for the organism to survive, it's very difficult to see mutations in these genes, because by definition, at least when they are completely missing from the organism dies. And so the important insight was really that there are now enough data that one can model the probability that different sites in the genome mutate. And that's really what you did, Gray, that you integrated a lot of data. And that's also, thank you to all the wonderful people who have shared all these fantastic epigenetic data sets. So you integrated these epigenetic data sets with mutations that we actually observed, which were thousands, which are a lot, but in the big scheme of things, that's still a somewhat small number. But using these sophisticated models, which you then, of course, validated in many different ways, you could then literally go to every base in the genome, and the genome has 100 million bases in Arabidopsis thaliana, and then calculate for every base how likely was it that it mutated? And that was really the key insight that allowed you to then look, do genes mutate at different rates, then sequences the outside genes, and then eventually do genes that are particularly important for the survival of the organism, do they mutate at different rates? And I think there has been quite some confusion when people only see the headlines. Of course, they automatically assume that this is all based only on direct observations, and this modeling was really an important part of it.
Yeah, that was kind of an aha moment when we realized that this mutation rate was so strongly predicted by the epigenome. And so for those who aren't have a biology background, when we talk about the epigenome, we're talking about the so when you look inside of a nucleus of a cell and you look at the DNA, you'd find that in our cells and in plants and animals, the DNA is wrapped around these proteins in these little balls, like beads on a string kind of image in your head. And on those beads, they can be chemically modified by other proteins. And so those chemical modifications on the beads on which the string is coiled, the DNA is coiled around, those marks are what we're talking about by the epigenome. And there's lots of data that describes how the epigenome differs between different places in the genome. So imagine, beads on a string, and the beads are different colors, and so you could look at it and say, and so it turns out that in animal and plant genomes, that the beads that are found within genes, so the places in the genome that actually code for proteins and the ones that are responsible for the most important functions, for example, you can think of them as in your head as like having a different color, like they just look different from the perspective of the epigenome. And so this is the critical insight that we realize allows for those genes to be recognized and receive like beneficial reduction in mutation rate.
Do you take a question?
Yeah.
So I just thought about this as I was listening. I joined maybe for the last 5 minutes, but this is something that I developed an intuition, a natural intuition, and I really like topics like this. So in terms of, obviously, because I'm not in the field, I will just use analogies rather than.
Yeah.
So I'll use two analogies. The way I see it
Mhm.
is genes can really biology, but biology can impact quite drastically. To play with what you just said, someone is breathing into the microphone very loudly. Right. So to
Yeah,
play on
yeah.
what
So,
you
so
said
what you just
earlier.
said. So genes are biology. That then the biology affect genes again. I think this is one of the super fascinating things about biology, that a lot of it is recursive, and I think that's totally spot on. And that's really in key insight in evolution, that there are always these feedbacks.
Yep. So it's like
Right.
I think I like the quote by the famous evolutionary biologist Dobzhansky. He says that nothing in biology makes sense except in the light of evolution. But I think when we think about mutation, it's also true that nothing in evolution makes sense except in the light of biology. So understanding the biology of the genome is also critical for understanding the evolution, especially when it comes to mutation, I think. So I think you're spot on with your intuition.
Right, right. So I'll have the question is basically I'll try to leverage into two analogies. So one is let's consider biology top down, right? And genetics bottom up, to use a psychology slash psychiatry kind of analogy approach. But here let's play with this analogy and tell me if I have the wrong intuition. But in terms of random mutations, let's use a political, but only as an analogy, not that I'm talking about politics, just like a political or geopolitical analogy in terms of understanding, let's say, the totality of our being, being organ, like micro affects, like genetics. How could that impact the totality of the being? So this is the globalism, and then localism. So sometimes not everything, let's say, is in direct communication, like a gene mutation and the purpose, right? of the totality of the being, the entities that we are. So because of that, it will make local changes, not maybe even like adapting. You know what I mean? So for example, if the mitochondria is exposed to high energy intake, it will spin out of control, so much so that at least as the cell is concerned, it's just healing. But it could spin so much so out of control that it will mutate and replicate itself, until you get zombie like or cancerous cells. So how could we, let's say, reduce gene expression or even gene mutation locally rather than just in terms of what our function is? Like, for example, a mole, HPV viruses. Could we reverse engineer viruses in order to reduce gene expression or gene mutations?
Yeah, I'll try to answer. So I think, I mean, yeah, I guess one way I'm kind of interpreting the metaphor is like these sort of alternative, versions of historical assessment, trends and forces versus, personality of individuals. And I think, what we're sort of sensing is that there's a lot of in the genome when it comes to mutation, there's the global scale, the sort of trends and forces are that, genes that are expressed a lot tend to get mutated less because of this general phenomenon. But can we, I think the question is, if I'm understanding, is like, can we instead sort of leverage our understanding to then go and manipulate the mutation rate of specific genes and reduce them? Is that what you're getting at?
My approach was more locally in terms of our body. So our body, let's say our body is something global and
Yeah. I see
certain genes. Yeah.
I see. Yeah. I think that if we understand the mechanism of how genes are preferentially repaired, then I think that it's not implausible that it would be possible to develop interventions that could leverage those mechanisms to then preferentially change the mutation distribution in specific tissues. I think that's probably within reason. I don't think that it's like imminently doable because I still think the mechanistic understanding needs to be advanced before we can take advantage of that. But I think our understanding of the way that DNA repair preferentially recognizes and fixes certain genes is like growing constantly, especially in the realm of cancer genomics. People are making incredible advances there about understanding how that works. I don't know. What do you think, Detlef?
Sorry, had to find the button to unmute myself. Yes, so one of the most fascinating aspects of your discovery, Gray, is really at what scale this changes. So coming back to what we just heard. So initially, through what you said, mutations are major fuel of evolution, and it's the mutations that actually led to mutation bias to evolve. And for me, one of the most interesting questions is how quickly does that change? So how quickly does a gene experience lower mutation rates, and how quickly can it perhaps revert to higher mutation rates again? And at what timescales can this happen? And I think the most fascinating question is, can any of this be regulated by the environment directly? So adaptation, over the span of generations, tens or hundreds or even thousands of generations, is something that we can easily imagine. But what if this was due directly to environmental factors? And this, of course, sounds a little bit crazy, but we know that, for example, the human immune system, it relies very much on very localized mutagenesis. And that's clearly stimulated by the environment. Depends on what pathogens we are exposed to, that leads to turnover in the immune system, that leads to activation of these pathways. So I think it's not an extreme stretch to think that this might pop up in other areas as well, although it probably is more subtle there.
Yeah, I
Another
agree.
brief question, if I may. I really like your intuition, Dr. Detlef, about how you correlated it with the immune system. And I have a question for the both of you, please, if I may indulge with my last question. It's, is there any correlation the
Yes, thank
way
you.
cells act, in the way that cells act and genes express themselves? For example, let's say the Y receptors in the nose, right? Is there any way we could, let's say, spread or even try to try to find mutations that we could engineer in order to like sprinkle, basically not to over energize mitochondria, you know what I mean? Not to allow mitochondria to spin out of control. Is there any way we could have a chain reaction and, you know, to spread all that high energy intake that might translate into mitochondria hyperdrive?
I think the simple answer is that the power of evolution should not be underestimated. That almost anything can evolve. It just depends on how long it will take. I mean, if you compare, life on Earth as it is today and as it was 3 billion years ago, if there had been an observer 3 billion years ago, who would have predicted that the surface of the Earth would look so different today. So, I the easy answer is that evolution can do almost anything. It's just for us humans, essentially impossible to imagine what that will look like because there are so many different paths for evolution to go.
Yeah, I think, yeah, along the same line, I think we should. I think that there's biology is in the same way. Biology is incredibly complex and elegant, and I think that this is why I think understanding how the biology of the genome works is critical. So for those types of ideas of leveraging, trying to engineer things, I think, yeah, understanding how things work and how they evolve too is also important, because understanding how they evolve tells us something about how they work as well. So studying both the mechanisms of genome biology and the evolution, I think, are important for even sort of thinking about those types of applied approaches.
So if there are no immediate questions, so first, thank you for these questions and these interesting ideas. If there are not immediate questions from the audience, Gray, maybe we can discuss a little bit the direction that this work could go, looking at mutation bias, not just in Arabidopsis thaliana, but in many different systems.
Yeah, yeah. I mean, I think what you said earlier about the environment is I totally agree. I think thinking about how the environment can change mutation rates in different genes is really interesting and has a lot of cool evolutionary implications, because if what we found is that mutation rate is affected by which genes are getting turned on and off, then it suggests by sort of deduction we can arrive at the hypothesis that, like, if the environment changes and genes are getting turned off or getting turned on, their mutation rates can change. And so for evolution that's quite cool, but also for like human health and other things, because we know that mutations are the source of things like cancer, and so understanding how whether they happen more often than not as a product of the environment could be really interesting. I don't know, what else are you thinking, Detlef?
Yeah, I mean, we already know that the environment, both in the body and also the outside environment, affects mutation rate. So not necessarily the mutation bias that we've observed in Arabidopsis thaliana, but certainly the spectrum of mutations. So, for example, in cancer, which you alluded to, it's very clear there are these different signatures of mutations. And so you can actually, in part, tell from the signature that a cancer has what was the original tissue it came from. And that's almost certainly due to the different epigenetic landscape and the activity of genes and in different tissues. In Arabidopsis, there's nice work from Nick Hubbard, for example, that when you stress plants, that mutation rates generally go up. I think there it would be super interesting to ask, if you stress the plants in different ways, whether there is a direction in which mutation bias, for example, changes. So just thinking about this ratio of mutations in essential and non-essential genes, can that ratio be shifted by the environment? I think it would be really surprising if the environment wouldn't shift this balance.
Could
And
you
of
speak a bit about high energy intake, maybe in cancer? Is there, findings about high energy intake being correlated with cancer or mutation, cancerous mutation?
I'm not familiar enough with that literature, so I just read something super interesting that sugar, for example, can sugar metabolism can affect mutation rate. So I think that would be sort of related to what you were just saying.
Yeah, or I mean, certainly like high energy I mean, obviously we know that, sunlight, high energy UV radiation can damage DNA. So that's obviously a mutagenic, you know, um, that's high energy, basically solar radiation can be mutagenic. So I think that's known. And I think that different types. So I think yeah. So it's been shown also that different types of DNA damage lead to different types of mutations as well.
A quick follow-up question, and this is actually a personal curiosity, but I think it's helpful for the community as well. Could you bust the myth of, the basically until recently, fairly recently, the consensus in the academia, in the academic world, was that us human beings, we inherit 100% of the mitochondria from our mothers. Could you bust that myth one more time?
I Yeah, is that yeah, I don't know. I'm not familiar with the literature there that's been disrupted. I mean, biology is always full of surprises, so I wouldn't be surprised if there was some deviation from 100% of anything, but yeah, I don't know. Detlef, do you know more about that?
Yeah, I mean that's one of the that's one of the things that for scientists makes it always difficult, because you can never exclude anything. So just because it has not been observed doesn't mean it can't happen. So again, I'm less familiar with animal genetics, but I don't find that entirely surprising that sometimes mitochondria from the father get transmitted as well.
Yeah, there was a paper, I think like a couple of years ago, that showed that in very few cases, exception cases, but that's all I also know about that.
Yep,
Hmm.
yep.
I, I
Yeah, and
actually,
I think
sorry.
there are some plants also where that happens, but I don't have that at the right now at the tip of my
So
whatever it is.
wouldn't it be a massive insight into like for example things that could spin out of control, such as mitochondria correlated with high energy intake? Like wouldn't it be a good predictor and indicator at the same time of someone being predisposed to cancer by knowing more about the mother?
I don't know. I don't know.
I have an answer for you, Andrew. If you let me, I mean, Katrina, are you fine that I can respond to Andrew? So,
Yeah, respond
sure.
really quick.
So, in a case of the mitochondrial, so in a certain age, for example, 20, 30, I mean, by increasing the age, we're going to lose I mean, huge amount of the mitochondrial function. And the main one is the NAD plus as a component, because we know that inside the mitochondria we have a different complexes, because you talk about energy mechanism, and mitochondrial is necessarily for the oxidative reaction, and we are familiar with the ROS, which is the we know that it's damage the cells and the function of the cells. So for example, it might be when you are 30 years old or 20 years old, you're gonna lose something, I don't know exactly the number, 25 kilogram of your NAD plus or NAD, which is very important for the mitochondria. So when we want to just, find out the significance or relationship between the mitochondrial loss or, kind of susceptibility to the cancer or such a thing, it can make sense. I'm sure that you can find lots of paper out there that they had a conversation around that.
Is there a correlation with increasing NAD and nicotine smokers?
It's a little bit off topic. Can we? I mean,
Yeah.
yeah, there's a correlation between like nicotine. I'm not sure, but NMN and stuff. That's David
Yes.
Sinclair's lab research.
It's
But maybe
deterioration.
we'll
It's
let
gonna decrease.
LT ask her question now, and then we move on.
Sure. Can I ask my question then? So I had a question about, death left. Am I right?
Mm-hmm. Yeah.
So my question is about the rate of the mutation, because as much as we have an expression, we have less
Mm-hmm.
mutation, as you just mentioned.
Mm-hmm.
And when we want to compare the brain cell versus somatic cell, so what is the number or the range that you can just give it to me for both of them in comparison with each other? This
Yeah.
is my
So
first question.
yeah. A big impact in mutation is, of course, during DNA replication. So that's pretty well established. And um, when we think about mutation rate, we both think about mutation rate from one generation of an organism to the other generation of an organism, but also the number of cell divisions. And so, for example, in the plant that we study, Arabidopsis, we assume that there are about 30 cell divisions from one generation to the next. And
So
so you
it
can
means 10 power what number?
we find from one generation to the next something on the order of 10 to the minus 9. Then you can, divide that to by, sorry, 10 to the minus 8. You can divide that by 30. So then you are 3 times 10 to the minus 10 or so per cellular generation. And so these mutation rates, they are not wildly different between different organisms. There are some organisms that have exceptionally low mutation rates, but most have mutation rates that are in a similar ballpark. And so that would mean for human cells that there is one mutation approximately for every round of cell division. And so that, works out quite well. So I don't know exactly what is the number of cell divisions that, for example, our germline goes through, but the literature is that we inherit from our parents, or mutations that happen in the germline of our parents. It's on the order of 50 to 100 new mutations that we have that our parents didn't have. So I think is really super interesting what you ask in terms of brain cells versus other cells in the body, because most brain cells do not divide. So do they? Is that an advantage, or is that disadvantage? I haven't thought about this. Gray, what do you think? Is it an advantage or is it a disadvantage not to divide in terms of mutations?
Well, I think you're right that because DNA replication is one of the main sources of mutation, because it's an imperfect process that when DNA is trying to replicate, new base pairs are being added, and sometimes the wrong one gets added, and so that can cause a mutation. So if in the absence of DNA replicate cell division and therefore DNA replication, I would suspect that the mutation rate would be lower. Though I'm not quite qualified to speak to the mutation rate in brain cells versus other cells because I just don't know. But it seems like from a mutation perspective, there would be some advantage of being sort of quiet from a DNA replication
So
Yes, we
so
perspective.
have
so,
more
so
express there. I know that we have more express in the brain cell, especially it's late in the cortical area and it's more in a
mhm.
subcortical area. That's why, I
Yeah.
mean, the
Yes.
mutation is less.
Yeah, I recently asked somebody who works on DNA repair, on the mechanism of DNA repair, and specifically transcription coupled DNA repair. And
Yeah.
I asked him whether, what's the balance? So transcription is mutagenic because when there's transcription, the DNA is unwound, and single-stranded DNA is more vulnerable to mutation. But then also there is transcription coupled repair because if there are mutation, it stops the transcription machinery, so, the cell has evolved mechanism to, when that happens, to recruit DNA repair machinery to fix this error so transcription can continue. And so anyway, so I asked the specialist, what's the balance? Is transcription, what's the net effect on mutation? Does transcription cause more mutation, or does it help to fix more mutation? And so he didn't know. Gray, have you looked into this literature whether transcription on balance is more likely to favor mutations or to reduce mutations?
I think it's hard to actually say because transcription itself, as you said, is probably mutagenic because it causes everything to be single-stranded for during transcription. Though I think that the levels of the levels of complexity are very deep because transcription also promotes different epigenomic marks, and those marks might
Mm-hmm.
be serving as signals for other repair. So in addition to direct transcription coupled repair, that are things that are repair mechanisms that are sort of riding alongside replicate DNA transcription, or yeah, transcription itself, you also have the addition and sort of enrichment for epigenomic features like these histone
Mm-hmm.
modifications, these chemical marks that I was talking about, that these are increasing as a product of transcription as well, which are recruiting other repairs. So I think it's like there's lots of interacting parts going on, and yeah, also DNA repair is it's a very complex. There's just many different types of repair. So there's many different ways that repair can happen, and I think that as we understand that stuff better, we'll be able to, the relationship between gene expression and mutation, I think is going to become clearer the more we learn about all of these
Yeah.
different molecular processes that are happening.
I mean, one of the things, Ray, that is actually surprising. So, as I alluded to earlier, there are some species that have really low mutation rate, and I think it's generally accepted by evolutionary biologists that mutation rate is not as low as it biochemically could be.
Right.
So,
And
there
also the
hasn't been.
percentile to the mass, like whales.
No.
Right.
In terms of so mutation rate, I think theoretically could be something like 10 to the minus 11 or so, but in practice, it's more in the range of 10 to the minus 8 to 10 to the minus 8. So actually, 2 to 3 orders of magnitude higher than what biochemically should be possible, which
Right.
is quite remarkable.
Yeah.
You mean
Yeah.
eukaryotes or prokaryotes? Which one?
I think anywhere.
Between the
Okay.
two, I think across all the sort of tree of life from eukaryotes to prokaryotes. I think
Hmm.
in generally, I think what people have found is that in prokaryotes, and so in bacteria, they have a lower mutation rate generally than eukaryotes. They have much smaller genomes, and so that's, I mean, this is a question. This is something that people have been interested in trying to answer for quite a while. Is like, why do we see differences in mutation rate between different species? Um, and there's been a lot of cool work there. But yeah, it raises some interesting questions about why is the mutation rate higher in species than it could be otherwise. It could be lower, but it's not. So that's kind of an interesting question.
Thank you.
Thank you.
And I think there also comes this this idea of recursive events that or recursive phenomena comes in there again. So the organism obviously can't look into the future, right? So the organism cannot quote unquote know what mutation will be favorable, but that concept is, as I said, is sort of demolished a little bit when you look at things like enhanced mutagenesis in the vertebrate immune system. So obviously, the vertebrates have quote unquote learned through evolution that there are regions in the genome where it helps to increase mutation rate.
Right.
I mean, it's so great. This is actually, so I have to admit that I hadn't, before your work, I hadn't really, thought about this a whole lot. But in hindsight, I find it actually somewhat surprising that, quite a few people reacted sort of allergically to this idea of mutation rate varying along the genome according to functional annotation of genes, because again, the vertebrate immune system is such a super prominent example. Do you,
Yeah.
when you talk about this, do you ever bring this up, Gray?
Yeah, it does come up. It does, yeah, because that's a really beautiful and well-known example of how so in immune genes, in immune cells, there's this very targeted mutagenic process. So clearly, like biology and evolution can find solutions that deviate really strongly from this sort of a random mutation. I think that the kind of allergic reaction from some people about the idea that mutations aren't random is I think it's sort of just a historical. I think it's a product of the history of the way that mutations have been spoken about for the last century, basically. Where I mean, I think the title of this clubhouse is something about mutations being not random, which it sounds kind of controversial, but in a way, it's almost an obvious thing that if random mutation, which is something that has been said for so long, I think has been a substitute for we don't understand why some mutations happen more than others, so we can call them random. But now our understanding of the biology is just so much richer than it was a century ago when those kind of phrases got stamped into textbooks and have been passed along, that now it's just sort of it's almost just a little outdated for us to just sort of describe things as random when we know what's going on. It's not a there's nothing mystically random about mutation. They're governed by molecular processes. So I think it's some of the reaction I think is largely from just trying to we have to sort of get past like a lot of like the terminology around randomness and mutation. I think some of
Yeah,
it's just I think
and Gray, I really love this analogy that you came up for Science Friday, where you
oh
said, it's kind of like a car factory. So it totally, makes sense that in a car factory, of course, errors in the assembly and the making of a car, that they will happen anywhere. But it makes perfect sense that the car manufacturer pays a lot more attention to any errors or mistake in the engine of the car than some, whatever plastic doohickey in the trunk that you can't really see anyway. So,
right
and why shouldn't it be the same in the genome?
yeah
It just said, and that's really is the key insight of this paper that we just published. So in hindsight, I know that also how we advertise it in terms of mutation rates varying between essential and non-essential genes, so not being random in that way. This was always front and center. But I think really the key discovery is that the cell has found a way of marking genes that are particularly important, so these epigenetic marks, and therefore that the cell can recognize all these genes as a class to be different from other genes. And that's again, we come back to what you said in the beginning. That was this original observation, and I think there would have been no reason actually to come up with this hypothesis that essential genes are somehow marked differently than non-essential genes. So when you think about this ideal of how science progresses, with specific hypotheses, in this case, it was really much more having an open mind and yeah, just letting the data speak for themselves.
Right. Yeah, totally. Yeah, I don't think we would have come out this expecting to find what we did. It's just sort of, it's
Hmm.
one of those fun kind of scientific experiences where you just stumble across something that's really compelling, and
Yeah.
then it pulls
I
you
mean,
in, you know.
although I think what's also, it's one thing great that I kicked myself a little bit that we didn't mention that in the paper, that when we talked about the vertebrate immune system, but there's actually really nice work in a number of other systems, so especially plant pathogens. And I see, one of our colleagues, Nick Grunwald, is here in the clubhouse session. He's working on these. So in the plant pathogens, for quite a while, there is this idea of what's called the two-speed genome, where there are parts of the genome that evolve faster. And those parts of the genome often contain the genes that the pathogen uses to attack its host.
Yeah, we
Yeah.
had also Dr. Krasileva here, Ksenia Krasileva. She said
Hmm.
she knows you too. I don't know,
Hmm.
from Berkeley.
Hmm.
That
Yeah,
also
yeah,
works
yeah, yeah,
on
yeah.
the,
She
yeah,
was
she was,
she was on your she was with you, what, like a week or 2 weeks ago, right,
yeah,
Ksenia?
yeah, a week ago.
Yeah,
Yeah, exactly.
yeah, 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 yeah. And
On the day
she
when
does
the
really
war
super cool
started.
work.
So yeah, it was a bad coincidence. It was the day the
Hmm.
Russians attacked
Mm,
Ukraine. So she was kind
mm.
of, worried. But
Yeah,
yeah,
it was probably a
I
difficult day for her. Yeah. Hmm. Yeah. So anyway, so once you allow yourself to accept to accept what we discovered, and then you look around and say, oh yeah, there is actually quite a bit of other evidence that, not exactly the same, but evidence that really goes in the same direction.
Yeah, I think it's almost sort of, once you sort of take a step back and you look at it, it's like almost obvious, that this would evolve and exist, and it's not so, which is kind of one of the fun, that's sort of was well, thinking about this has been helpful because it's sort of a sanity check. It's like, okay, this isn't that crazy. It's actually quite makes perfect sense, so it's kind of nice to be able to look at all these other cool examples that have been described, where people are finding other examples of biology where it's found solutions to mutation that lead to these cool non-random patterns as well. So it's always nice to like see that, to feel like, okay, we're onto something that's consistent with what other people are finding as well.
Hi, everybody. And Detlef, thanks for mentioning the two-speed genome hypothesis. So I have a question for both Gray and Detlef. I'm wondering if you hypothesize what the mechanism might be for maintaining that mutation bias, and maybe opening the door for evolution to occur when a population is stressed, or when the environment changes and the plant community needs to adapt much faster, or I mean the population needs to adapt much faster. Any
Yeah,
thoughts on
yeah,
that?
I think, Nick, I think this is definitely the next direction to go, to study at scale, both what happens in different environments in terms of global mutation rate, but then also mutation bias, and then really the genetic underpinning. So I think the genetics is going to lead the way. And in our paper, we had already, there was already some indication that there's a genetic basis, because we looked at spontaneous mutations in different strains of Arabidopsis thaliana that come from different places in the world, and there were clearly differences in the mutation spectrum. Now, finding these differences does not necessarily mean they are adaptive. So, a lot of things vary in nature without them necessarily being adaptive. But even if they are not adaptive, if there's genetic variation, if we can find the genes responsible for this variation, I think that's going to be a really nice entree
Yep.
into the mechanism.
Yeah, I agree. And then as for the mechanism, I think we can arrive at some pretty compelling hypotheses by looking at what's known in humans, for example. So in humans, we all have a mismatch repair gene that has a domain in it, a protein domain that recognizes a certain histone modification, and that's what leads it to preferentially bind and repair gene bodies in human cells. And so that's a really compelling case of what's possible. That you can have repair genes that have domains that recognize the epigenome, and that serves as a mechanism of preferentially repairing certain regions. And we've looked in plants, and it seems to be different, but I think it gives us a good clue about what might be possible. And then as far as responding to stress, I think that leads, yeah, that's an awesome idea and question. You know, because the epigenome itself can shift as a function of the environment, then it leads one to wonder about if it's under stressful conditions, how does the epigenome change and therefore shift the mutation bias? So, I mean, these are all just awesome questions that I'm looking forward to seeing being answered in the next few years or so.
Yeah. So some of the most exciting ideas in that direction actually come from transposon biology, and of course, transposons are very relevant to this two-speed genome. So, a friend of mine, Jurek Paszkowski, a number of years ago, they had found that there are transposons that have elements that are heat responsive, and they call these transposons onsens, for the Japanese hot springs. And so what they proposed, I think, in the end, they couldn't show this directly because they weren't able to do the experiments long enough. But I think super compelling idea. So plant is under heat stress, then these transposons are mobilized because they have heat response elements. They can then insert somewhere else, and they can make other genes heat responsive. And so making other genes heat responsive could be helpful in adaptation. Now, depends a little bit how these transposons insert. Do they, insert entirely randomly across the genome or not? We know that, transposons generally do not insert entirely randomly, so you also have to take that into account. But I think that would be a really nifty mechanism how to translate an environmental change into making the gene complement in a genome behave differently and specifically tailored to that environmental change.
Yep.
I want, so Dennis, you came, and LT, you came to the stage. Do you have a question?
Thanks, Katerina. Yeah, I did have, it's maybe a little bit of a high level question, but so given that there are 3 billion base pairs in the human genome and there's an error rate, I don't remember what it is. And for example, in a giant sequoia genome, there's 26.5 billion base pairs. So just in terms of, how many base pairs are in this organism, and is there any sort of error rate that you were able to look at, or if you had even considered it? I understand it might be outside of the scope.
Yeah,
But
yeah. So actually, what's sort of interesting is that so you might think, so what happens if you have a much larger genome? So actually, when it comes to eukaryotes, eukaryotes have a relatively small range of genes. So it's normally on the order of 30,000, give or take. So it can be 20,000, can be 40,000, but so ballpark is 30,000. And the size of proteins is on average pretty similar. So the coding, the portion of the genome that codes for proteins, that actually doesn't change a whole lot. So it's remarkably similar between Arabidopsis thaliana with its 100 million base pair genome and corn or humans with 3 billion base pair genomes. So that means the mutation rate per base pair scaling is perfectly fine. So it doesn't have to be, any lower the mutation rate in a human or maize genome. The chance that it will, hit a gene and change coding sequence will actually stay the same. And that's why there is actually not as much variation in mutation rate as one might think. Great, did I get this about right?
Yeah, that sounds about right. I think like as genome size scale, so the mutation rate generally, it's a per base pair unit, and I think across different genome sizes, I'd have to check, but I think there's certainly differences between species, but I don't think it doesn't scale with genome size in a way that's perfectly predictable or linear. I think that so the result is that if you have just species with larger genomes like giant sequoia, they have more mutations, total number of mutations, but on a per base pair basis, it doesn't increase nearly as much as, or decrease nearly as much as it does
Yeah.
the
So
relative.
so they have much more DNA that doesn't really matter a whole lot, and therefore it's fine if they have a lot more mutations total.
Right. So like the per gene, in terms of genes being the regions that code for stuff, these the per gene mutation rate is similar. Um,
Exactly. Exactly. Hmm.
Thanks for that answer. It's actually kind of interesting considering that they do live 2,500 years. So
Hmm.
I wonder if those
Yeah.
extra genes are doing
Yeah.
something we just don't know
Yeah,
about
so
yet.
so so that's that's actually something we're trying to get at right now. Gray, I don't know whether we talked about this, but there is the Napoleonic oak in Lausanne. So there have been a couple of papers, and so they are. I think the evidence is sort of out there whether in trees the per generation mutation rate is the same as in short-lived plants or not, and whether they do something special trees. Do they set cells aside that divide extremely slowly or not? So Gray, we're actually going back to this specific tree in
yeah,
Lausanne
that's cool.
and trying to see whether with better genome assembly we can get better estimates of what
Yeah.
mutation rates are.
Yeah, that's great. Yeah, I think for trees it's really cool because they have all these branches, and as the branches are developing, they can presumably be accumulating mutations, and so and people have done this now in trees, and you can sequence different branches in a tree, and you can see mutations that arose at sometime during development a long time ago, and you can see those mutations now at the tips of these branches, and so it's kind of
Hmm.
like a little evolutionary tree. Each branch is accumulating mutations. So yeah, it would be. I think that's going to be really cool. So
Yeah, there was actually really nice work from Sally Otto for quite a while ago, and this was very indirect. But she asked whether seeds from different parts of the tree, was there any evidence that basically they became less fit with time, and that really is what seemed to be the case. And then, of course, in humans, we know that older fathers, on average, have more mutations in their sperm than younger fathers.
right,
Okay, may I ask my question? Now it seems to be a little bit more stable. The question is that you mentioned about genome size and the mutation rate per basis. But then have you, I mean, of course, you're going to look at, besides the Arabidopsis, you're probably going to look at C. elegans and then Drosophila, right? Small little animals, plant besides other plants. And then the different organs. And one more thing I'm thinking is that, like certain genes, besides they're
Hmm.
clustered together, essential versus non-essential. It's
Hmm.
harder to find the essential genes because mutations because they're dead, right? When you do the
Hmm.
selection, you
Hmm.
cannot discover them.
Right.
Then the other question is that when you do the certain genes that turned on in the early on or genesis, spermatogenesis, and then later on they go on again, into later stages. So they have this developmentally regulated.
Hmm.
What about the rate in those two phases? Thank you
Hmm.
very much.
Yeah. So one thing so one thing that we do know that the timing of DNA replication that certainly matters in terms of when a gene is transcribed. That's also super interesting question, because almost certainly at different phases of the life cycle, the DNA repair machinery is going to be active active in different manners. I think that would be very cool direction to go into. I think well, at least my lab will stick with plants. I don't know about you, Gray, whether you have ambitions to look in especially the human data, but certainly the human data would be a treasure trove to look into, because there are gazillions of mutations known.
Yeah, I think we'll probably, I think we're always open for collaborating with people who work in other systems, but as far as what we grow in the lab, I think we're going to stick with plants. But some of these ideas and some of the sort of genomic comparisons do sort of lend themselves to looking across different species, and so we can look at the evolution of repair genes across the tree of life, for example, and that allows us to sort of expand in a general sense. But experimentally, I think plants provide a really powerful system because we can yeah, we can, they stay in place and we can do different genetics with them. I'm always happy to see, I'm really excited to see what the animal community does, and looking at, you said Drosophila and C. elegans and all these other great model systems. So I'm excited to see what comes out of that work as well.
In,
But yeah, just another point. I just wanted to address your question about essential genes, and if they're mutate, how do we know they're not mutating, because mutations in them would be lethal? And that's an awesome question, because that's obviously something that would prove problematic if all we were observing was kind of survivorship bias. Then it's not really interesting. We're just seeing selection and not mutation bias. And so one way that we can address this is we can look specifically at silent mutations. So we can ask as most mutations in genes are many mutations in genes are synonymous. So the change in DNA doesn't change the protein sequence, or they happen in introns where they're not affecting coding regions either. And so when we look at those two classes of mutations as well, we see that they are also much lower than expected by chance in essential genes than they are in other types of genes. So that appears to be the product of mutation reduction, rather than, you can't really, it's difficult to explain that as a product of selection, because we really don't think that the selection on synonymous changes or intronic variation is nearly strong enough to explain why they would be reduced as much as they are. I hope I hope that answers the question.
Yeah. I mean, maybe quickly, Gray, in terms of organisms to look at, what
Yeah.
I find really surprising that there hasn't been more work in bacteria, because they are, such a great system to do that. So I hope that our work is an impetus for people who work on bacteria to go back and measure mutation rate under a lot of different environments again, and in different species.
Yeah, I agree. I do think that there's differences that are interesting between eukaryotes and prokaryotes, though, because the fact that chromatin is so complex, and the epigenome is very, there's such big differences between the epigenome in prokaryotes and eukaryotes,
Hmm.
that we have these nucleosomes and these histone modifications and all of this elegant genome sort of chromatin biology that I think is part of the eukaryotic package of innovations that might explain that I think is different. And so I think we have to, in terms of comparing bacteria and eukaryotes, I think we have to keep in mind that they have very different genome biology. So
Right.
the comparisons
But
are
but
going to
I
be
but I could imagine that bacteria, came up with a solution that is different
yeah,
from what
sure.
eukaryotes came
Well,
up with. So.
that's what this beautiful paper by Martin Carrena in 2012, where they find a similar bias to what we find in some in a similar way in bacteria. And so they're suggesting that there's something in bacteria going on. So I think it's yeah, very much worth exploring for sure.
This is such a great paper. I mean, like four of them. Is it four related in the same journal,
Hmm.
in the same article? Yes.
Hmm.
And thank you. Everybody's questions are stimulating. I really totally enjoy it. Thank you very much again.
Thank you.
Thank you. Thank you.
Yeah, I wanted to check with you. It's an hour now. I think Prakash, he came up to the stage. Are you okay with taking one more question before we close the room?
Okay.
Sure.
Cool.
No problem. Happy to.
Okay, go
Yeah,
ahead.
thank you, Katrina, and thank you, Detlef. Uh, I know I've been following your work for a long time because we actually met in a Arabidopsis conference in the
I
90s.
know.
Yes, okay. So the question I have is, if you put Evo-Devo as a tool to further delve into mutation bias, I
Mm-hmm.
always think that we have if you go because I've been working with crops and other things, and most of the time the tools are developed around the concept of G by E. Uh, but I always ask the question, where is the D in the middle? So it's a G by D by E all the time. But the
Mm-hmm.
D is only derived in retrospection with reference to after you arrive at a phenotype.
Mm-hmm.
So the question, like when you have a concept like mutation bias, where do you see this leading forward in terms of kind of, if you want to, for a lack of better word, forward selection? That if you can, is there a prediction that one can go through when you understand modules and gene regulatory networks, and ask where are these mutation biases lying within these things that are so critical for survival? And that
Mm-hmm.
is being selected for in breeding. For example, in yield increase or phenotype of the inflorescence or the stability of the architecture. Can we kind of rest our resolution questions with reference to regulatory elements, open reading frames, or super enhancers, pioneer transcription factors? Is there a screening procedure that you're going to be working on? I'm kind of curious because I read your paper several times,
Mm-hmm.
but you had hinted on it towards the end, but I was just kind of curious where you're going to take this further. Thank you.
Well, I mean, one direction is certainly what we alluded to already. When you think about, differences in development in plants, it doesn't get much different than, say, sequoia and Arabidopsis. So that's definitely one direction. The very long-lived plants that have a very different body plan and architecture from short-lived plants. That's one direction to go into. I admit I hadn't really thought about this, whether this is a process that might have been affected by domestication and improvement of crops, but that's really that's a very cool idea. And Gray and UC Davis, what better place than there isn't a better place than UC Davis to think about this, whether there has been impact of breeding on this. Is
Yeah.
that something that you're considering, Gray?
I mean, we're certainly interested in thinking about these processes in crops, and thinking about how they affect crop variation. For example, if mutation bias reduces variation across a lot of genes in the genome, it raises questions about, for breeding, is this a constraint because there's a lot of variation that's missing because it mutate very often, and is this interesting or important for thinking about the origin of genetic diversity that's used in breeding and crop development, and then have systems been perturbed in through the domestication process? I mean, there's just so many cool questions for us to ask, and yeah, I think it would be quite interesting to ask these in crops. But also, to your questions about development, I do think that all of these mechanisms in the biology and the evolution, they are intimately connected to development, because the mutations that get passed on to the next generation are the ones that have to happen in cells that contribute to the germline. And so it becomes, you inevitably start thinking about developmental biology, of which cells in the meristem are accumulating mutations and whether or not they're going to get passed on to the next generation. And so there is an intimate connection, I think, between the sort of developmental biology in cellular biology with the evolutionary processes, because of the questions about what's passed on from generation to generation. So there's so many cool sort of questions that emerge,
Mm-hmm. Mm-hmm.
and I'm curious about. So yeah.
Mm-hmm.
Thank you, thank you. Just another point to that is that with the at the institute where I work, this pan genomes of wheat,
Mm-hmm.
10+1, and several genomes are being sequenced.
Mm-hmm.
So, is there a tool building exercise that will be going on to discern mutation bias across these pan genomes? Because that'll be the most constrained data set that one can screen for these kind of things.
Mm-hmm. Mm-hmm.
Because some of them are like, you can start with einkorn wheat or
Mm-hmm.
Triticum murata and then come all the way to aestivum. Not worry about the ploidy levels, but you can have the gene sets and the regulatory
Mm-hmm.
elements, and
Yeah.
then start building tools around it.
Right.
Yeah.
Yeah.
Thank you.
I mean, Prakash, I think this is also, certainly something that will be very much worth considering, whether, ploidy levels, has that affected mutation rate variation in any way, because there is relaxed selection, and does that translate also in shifts in mutation rate? I think that would also be super fascinating to study.
Yeah, I agree. So yeah, you talked about wheat, and there's different subgenomes, and there's some subgenome dominance, and asking questions about, do different subgenomes have different mutation rates, and what are the consequences of that for all for evolution and breeding? I think it's going to be really interesting. And so,
Mm-hmm.
I'm excited to see what questions people come up with and pursue. I think it's going to be really fun to see.
Mm-hmm.
Thank you.
Thank you.
Yeah, thank you so much for being here and answering all these questions. I had like no work because you even had to you even ask questions each other. So this was great, and I hope you enjoyed this too.
Yeah,
And
totally,
please,
totally.
yeah,
It's
yeah,
wonderful.
please
Thank you.
come back anytime with updates about your research, other projects. We are always happy, to have
Super.
you here or listen in when we have other guest speakers.
Of course, of course. Yeah, no,
Yeah,
I could kick myself. I only found out a couple of days ago that you had Xenia on, so I would have loved to listen to Xenia.
Yeah, well, yeah, and yeah, I will definitely come back. I guess you guys can see this. I have a little symbol. It's my first time on Clubhouse, so I'll be definitely back. This is really cool. Thank you so much for the invitation.
yeah, thank you. Yeah, we have tomorrow actually at the same time, Dr. Kalsa, he will be talking about anxiety that is associated with autonomic hypersensitivity. And we have many more rooms coming up. So, and Xenia, she said she will come back, and maybe some of her lab members will come back. So, there will be more opportunities. So,
Great, super, super, great. All right,
Well,
thank you,
okay,
everybody. Thank
thank
you so
you.
much.
Thank you.
Bye.
Thank you.
Enjoy the rest of your day,
Bye,
evening,
bye,
morning, everyone. Bye.
bye.
Bye, everyone. Thank you.
Thank you.
thank you.
Great room.
Thank you.